Hello, I'm Jiahua Wu from National Donghua University, Grade 2. Today I'm going to tell you what I'm studying in the last year. My title is The Study of Molecular Signature of Regenerating Aeolosoma Viride using two-photon fluorescence lifetime imaging microscopy. For first, let me introduce about a worm called Aeolosoma Viride. Aeolosoma viride is a worm that is about 2 to 3 millimeter, and it can regenerate itself asexually within 5 days. Because of this amazing regenerating ability, we choose this worm to check whether there is some component plays a major role in regenerating aeolosoma itself or not. And after knowing the worm and the purpose of my study, let's have a look on figure 1. In this spectra, we can clearly see some peak like CCH3, CC, and C double bond. And in the optical image, we can know from where the spectra has been taken. And for the spectra, we can know these 3 peaks means one component. The azancine carotenoid means it can be observed in this worm Aeolosoma viride. Then let's move on to figure 2. This is the image describe different status of Aeolosoma viride during the regeneration. But for this poster, we'll be just focus on 0, 24, and 48 hours after amputation. The other hours will be described in Peiyang Huang's poster. Peiyang Huang is the second author of this poster. If you have an interest on it, please feel free to have a check on the poster. Then for figure 3. Figure 3 are the Raman spectra that we measure the tail part of aeolosoma after amputating it. In 0 hour, some weak intensity in the spectra can be observed. We assume that it might come from the food because the intensity is very weak. Then in 24 hours, which means the blastema formation, we can observe the strong intensity of the azancine carotenoid. So we speculate that during blastema formation, large amount of ROS is generated, and the zeaxanthin component is generated to antagonism with it. In 48 hours means blastema proliferation. We can see the zeaxanthin components can still be observed. So we surmise that it's because the zeaxanthin components start to spread to the whole part of the body. Then let's move on to the last figure in this poster, figure 4. Figure 4 have 3 pictures. These 3 pictures are all expressing the fluorescent proteins. But what's the clear component of that shining spot? We are now endeavoring understanding it. So here is the summary of this poster. First, carotenoids in Aeolosoma viride was observed at 48 and 24 HPA. We assume that it plays a major role during blastema formation and proliferation. Second, Raman spectroscopy and two-photon imaging demonstrate its feasibility for detection of single cell in worms. In vivo two-photon imaging was possible without any anesthesia. This is the reference. That's the end of the poster. My name is Jiahua Wu from Donghua University. Thank you. Have a nice day.